chromosomal anomalies on the 23rd set. Did we not have XO written down in your notes?
XO, XXY.
XXY, XXX, and XYY is what we had wrote down in our notes. And we were looking at some terminology in the book leading up to this. Let me get turned to that page. And terminology was on page 84 and 85, and we talked about non-disjunction. Those of you that were here the other day, we said non-disjunction is what's causing the monosomic and trisomic problems. And an N minus one is a monosomic. We had chromosomes lined up, and then those chromosomes were supposed to separate and move back to poles. Well, non-disjunction, what happened is the chromosome didn't separate and it stayed with it on that side, so we end up with eventually 2N minus 1 over here and a 2N plus 1 over here. So this was trisomy, and this is monosomy over here. This one would end up with 45 chromosomes, this would end up with 47 chromosomes with that egg or this egg being fertilized. And that's the non-disjunction we had discussed the other day. We also said it was completely normal for us to polyploid plants, but that didn't work very well with animals. And the other terms we were looking at were on page 85. The term inversion. The term inversion is basically we rearrange, and it's not supposed to happen, but instead of reading the DNA this way, it flip-flopped the DNA. We read it the same way, but the DNA was backwards when we read it. So that really creates some serious problems. And then if non-homologous chromosomes exchange sections, that's called translocation. And that's what in prophase 1 of meiosis, when we had crossing over, I know we're having a reel back in our memory here. You remember when they had changed part of the left leg for a right leg? We had red ones and blue ones exchanging, remember that? Well, those were sister chromosomes. Those were homologous chromosomes. So they were exchanging out equal parts. They were exchanging out blue eyes for brown eyes. The eyes were still there. Non-homologous chromosomes in translocation, if they exchange parts, it's like I'll trade my left foot for your right arm. So now we have somebody with 3 arms and 1 foot. You know, so this is messed up, big time. And the next one is deletion, where part of a whole set of chromosomes just gone. And lack of information is way more lethal in animals than too much information. And then the last one is duplication. This is not as lethal, but it really messes things up when you repeat the same thing a couple times. So they duplicate too much information. Let me give you an example of a deletion one. There is a syndrome on chromosome set 5, and part of set 5 is missing, a deletion process, and it causes a problem called cry-do-cat. It's spelled chat, but it's called cry-do-cat syndrome. Cry-do-cat syndrome is a syndrome in which the baby is born with the head larger than the body proportionally than it's supposed to be, mentally retarded, may or may not live a year, and will die soon. But when this child cries, the cry sounds like the meow of a cat. So therefore the name cry-do-cat syndrome. Yeah, that's a deletion. We can have trisomy on set 13. We can have trisomy on set 18. Both of those happen. They're not overly common. It's like 2% of the population might run across one of these. Both of those are basically death sentences. Most of those don't make it to term or even born alive. Only a handful are ever born alive and die shortly thereafter. I know of one documented case where the child lived, I know he was 8 the last time I checked up on this child, oblivious to the world, had no clue what was going on, totally dependent on whoever took care of him, and at any minute his medulla was going to say, I forgot to breathe. I mean, there's so many neurological problems, that's the case. And so that was trisomy 13 or trisomy 18. There's no age-related things there like they claim with Down syndrome with the 40-40 process. 40% of women over 40 has a 40% greater chance of having Down syndrome. It doesn't seem to be related to the trisomy 13 and 18. Okay, now from that, let's just look at 4 different chromosomal anomalies that occur with the 23rd set. The 23rd set being your sex chromosomes. The first one is XO, and XO represents Turner syndrome. Turner syndrome is a female. Book has identifying, our book may not, but if you research this, it'll have identifying features or characteristics. As we see with most genetic defects, they're not all classic. You have different levels. Some are mild, some are major. This person, just across the board, is short in stature. So next time you walk down the street, see somebody short, you don't, oh man, they've got Turner syndrome. You can't just say that. It's not normal to do that. The other thing, they say they're below normal intelligence. And I actually knew a person with Turner syndrome that had one bachelor's degree and was working on another one. She wasn't below normal intelligence, I'm telling you. Another thing is a webbed neck. Now that's one of these, you can look up webbed neck and see. I'll look at it kind of like bodybuilders. It's got all this muscle that comes down with, instead it's kind of skin. So kind of a short neck and skin coming out to the shoulders. And this young lady did not have that. And I wouldn't have known she was Turner's, but she told me she was Turner's. She was diagnosed with Turner's when she was little. So, some books will say they cannot reproduce. There have been some cases where they have. So there's all kinds of exceptions to the rule, but we do know it's a female. The O represents we're missing an X chromosome. So this is a monosomic condition where we have N minus 1 or 2N minus 1, basically where we're at, 45 chromosomes. Here we have Klinefelter syndrome. Klinefelter syndrome is a male. It's a male because of that Y. That Y has the testis determining factor on it. It turns on the situation that causes testes to develop. We get testosterone, we get male characteristics. So we have a male, the 2 X's produces a little more estrogen than he needs. So he cannot reproduce. He has breast development and he has underdeveloped male genitalia. Outside of that, he's of normal intelligence, can live a functional life, but like I said, cannot reproduce. Now they found this person in the Olympics, and this person was running races apparently very fast. The problem was he was running against the women, and when they discovered that she was technically a he, they disqualified her from him from all of her races. There we go, I finally got it right. But how they discovered that was with a genetics test. What led them to a genetics test, I don't know. But if you're going to the Olympics, you'll do a genetics test before you ever get started now. Now, let's describe this person. He's a runner. What do you think body fat? Zilch. What do you think muscle on his legs looks like? Now, granted, he's probably got pretty hairy legs. Maybe he shaves his legs. Yeah, aerodynamics. That's what I'm thinking. Well, let's look at a female runner. What, body fat? Zilch. Muscled legs? Absolutely. Breast development would be minimal because a lot of that could be fat, so that's minimal. Well, what do we describe with him? Minimal breast development. Other than him being ugly, there's not much difference in the two. So, man, look at that ugly woman looking out there running those races. Other than that, they're pretty much the same. So there was just some genetic problems going on there. Now we can apply Klinefelters to cats though. Anybody have a calico cat? You like calico cats? You like calico kitties? Well, for a calico kitty to get the combination of colors to make a calico kitty, it must have both X's and the combination of colors to give it the calico cat colors. To be a male, it has to have Klinefelter syndrome. to get the combination of colors to make it the calico male that it is. The problem with that in cats is not only is that male cat, if it's alive, it's sterile. Most of them are stillborn or miscarried. It's not only sterile, but it's mentally retarded. And so I'm asking myself, well, what's the difference of this cat and any other cat out there? Because they all act mentally retarded to me. I had one named Stupid one time. You know how she got killed? Run out from a car. Now, how stupid is that? But she lived for years. I don't know how many bunch of kittens she had, but her name literally was Stupid. She came to Stupid. Here you have a meta female. What do you think those 3 X's does for her? You're afraid to say, aren't you? But you're grinning about it over here. Maybe a little extra estrogen than she might need. Maybe she's a little emotional. It doesn't really say anything like that in the book. The only thing, in fact, this person could live her entire life and never know she has this chromosomal defect. She can live a normal life. She can reproduce, normal intelligence, function in society. You never know. One of you in here may be this right now and not ever know it. The only thing they ever documented that might be different for this person is that her 28-day cycle run erratic. Well, in today's society, how abnormal is that? It's actually normal, so they don't even write it in the books anymore. They can't find anything different. Now, this person may have a son that's Klinefelter's, or could have a daughter that's metafemale. That chance would increase. But other than that, she could live a normal life. Okay? Then this one down here, I waited to write this out so you'd pay attention to the other ones, because usually I lose the girls' focus on this immediately, because this is a super male. Usually they focus on this one and they don't hear anything I say up here. I'm telling you, it's not worth focusing on. Guys are messed up anyway. And I already told you all the day you get married, the day you start raising your first child, right? Did I already tell you that? Well, it's the truth. And I tell you the truth because, thank you very much. See these 2 Y's right there? That's way more testosterone than any male should ever have. This is a male on anabolic steroids, and he's not taking any drugs. His body's producing more of it than it should. He could live his entire life and never know he's a super male. There are some things that click along with this, though. If this person carries the gene for baldness, baldness is sex influenced. It's influenced by testosterone. If a male has a bald gene and testosterone, he's going to go bald. Okay? Now, are they bald females out there? There are. They have to have both bald genes, and testosterone's not going to influence that, but both bald genes show up and she becomes bald. But that's kind of rare, isn't it? See a few. All right, so in this guy's case, let's say he has a bald gene, and at puberty, he starts going bald. I mean, he like wakes up one morning, there's blotches of hair on his pillow. The hair just starts falling out. That would be a telltale sign something's wrong. You think mom's concerned when she picks up hair out of his bed? That's something, what'd you do to yourself? Well, I really had a rough dream last night. I pulled all my hair out. That wasn't it. His hair is just falling out. And so mom takes him to the doctor, concerned about this young man, tells the doctor all the symptoms of things, and he's going, does this bother you? Well, then she switches doctors because she realized she was at a psychiatrist instead of a real doctor. He runs a genetics test, and when he runs a genetics test, he comes back with the results that he has this genetic configuration. So he sets mom down, and he says, "Ma'am, your son's a Superman." And she said, "Well, thank you. Thank you very much. He's a good boy." But what does that mean? How long does he have to live? That's how bad it could be. And then he goes on to tell her, his body's producing more testosterone than it should, which is going to increase his chances of liver cancer and or testicular cancer somewhere down the line. It's no different than anybody taking anabolic steroids out here that shouldn't be. It's no different. So he's not diagnosed with cancer at this point. So he's thinking, doctor's going to make a recommendation. Say this kid's 14 years old. Doctor recommends to mom, we can stop this chance for cancer. We can lower his chances for cancer back to that of a normal child if we remove one testicle. Now, if the kid heard that, he's already jumped out the window. He's not, you just leave me alone. Mom's thinking, if it'll keep my son around, we'll sacrifice part of his body. But if you think about it, that lowers testosterone levels, doesn't it? Could get it back to normal, probably will. Could he go on and reproduce? Sure. So really it puts him back to normal instead of being the linebacker that he shouldn't be. Football scholarship, Dad's in there, leave him alone, he's got a football scholarship. So it'd be brain damage one of these days, if we get him through school. But that's your super male. Now there's another aspect of this. There was some psychiatrist in a prison one time, and they was doing some research in this prison. Why were these prisoners in there? And then I'm not sure what their original research was, but as they was looking through these files on these prisoners, they found that 5% of the male prisoners in this prison had super male configuration. And they thought, so a psychiatrist gets the old brain running here, and they think, here's our hypothesis. These guys are in here because of extra testosterone. They're in here for sex crimes. They've been raping people. They just been mugging people. That's their hypothesis. Extra testosterone, sex drive, kind of makes sense. So then they went and looked further into their charts and interviewed, and they found that it wasn't the case. That same percentage of these guys were off 7-Eleven as the normal population. I say normal. Is there a normal population in a prison? Normal genetic people in a prison, put it that way. So they's out there robbing 7-Elevens, and a small percentage of them's in there because of rape, and a small percentage of these raping, and another percentage is robbing 7-Elevens, no significance there. So that hypothesis didn't hold up. But what they did note, and this wasn't even part of their hypothesis, is that all these guys had anger issues. Extra testosterone, they were a little quick to fly off at the hammer, and they did have some anger issues, and that still stands for today. So if you know anybody with this syndrome, they have anger issues, according to the research. Any questions over these? These are not in your book, so good thing you wrote them down. But I think they represent chromosomal anomalies pretty well, and they're pretty unique and something we can apply to every day. No questions? All right. The rest of this chapter that I want to knock out today is dealing with DNA, RNA, and proteins. DNA, RNA, and proteins. There are 3 terms that pertains to this. Replication, transcription, and translation. Here's your definition of these 3. Replication is DNA making DNA. DNA just simply making more DNA. That's good. Your cells need to replicate, so. Transcription is DNA making RNA. DNA making RNA. And translation is DNA making proteins. We're actually translating proteins from the RNA that we just previously made in transcription. Okay, let's do a little bit of background on DNA and RNA, and then we'll go into the process of these 3 things happening. And yes, you're going to have to recall back to earlier chapters when we talked about the cell cycle and mitosis, and this is all part of it. Who discovered the structure of DNA? Watson and Crick. Thank you very much. Watson and Crick discovered the structure of DNA. They researched on this obviously for several years, and they were both scientists. One was a physicist, the other was a biologist, and they took a leave of absence from their respective colleges to work together on this. But from what I've read and heard, by the time they get up in the morning and take a morning walk with their dog and drink a cup of coffee, it's lunchtime. Then when they eat lunch, now they're sleepy for a nap, and they might spend 2 to 3 hours a day on this. Boy, I know for sure that was a government grant. And they've led this for a long time, but what they didn't know is there were 2 other scientists working on this named Pauling and Corey. You don't have to write those down. Some books have them in there and some don't. But Pauling and Corey was working pretty hard on this. And they're the ones that wrote the first document on the structure of the DNA molecule. The problem is they said DNA was a triple helix structure. Well, as soon as Watson and Crick read this, they knew that wasn't correct. They was pretty darn sure, but they needed to prove it. All right, so Watson and Crick used basically three different people to help them. I'm sure they had a bunch of other people, but there was a guy named Maurice Wilkins. Maurice Wilkins described the backbone of the DNA, which is sugars and phosphates. Okay? And then you had Erwin Chargaff, who described the bases. And the bases are adenine, thymine, cytosine, and guanine. Adenine and thymine always join together, and cytosine and guanine always join together. And that's Chargaff's rule. And it gets technical in the book, and maybe even in your notes. I might have said something in there about the purine. Adenine always joins with pyrimidine thymine, and the purine cytosine always joins with pyrimidine guanine. So that's Chargaff's rule. But the one that really turned the corner for him was a lady named Rosalind Franklin. Rosalind Franklin's specialty was X-rays. Now, in 1965, Watson and Crick got the Nobel Prize for the DNA structure. It was either 66 or 67 that Rosalind Franklin was up for a Nobel Prize with her work with X-rays. She died that year with cancer at 37 years old. Now, we know a lot more about X-rays today. She was sacrificial in that effort. She had no idea. Can you imagine the amount of X-rays she's bombarding herself with every time she take a picture? But she was able to take a picture of the DNA molecule to where it actually cast shadows, where they could see the front, and the shadow showed the back of this DNA molecule, and it showed a double helix structure, 2 sides to it. And from that, Watson and Crick used these pictures, wrote their rebuttal article, and Pauling and Corey read it, looked at the pictures, and agreed they were wrong, and Watson and Crick was right, and that's how Watson and Crick got the Nobel Prize. Off that government grant, I'm sure. All right, so with the work of all this, we had adenine, thymine, cytosine, guanine, and we'll do a thymine and adenine. Okay. So we have a DNA structure here. We have a sugar, a phosphate, and a base. What is that? What in your notes has a definition of a building block of a nucleic acid containing sugars, phosphates, and bases? Nucleotides, that's right. So that's a nucleotide. That's a building block right there. Another building block, so you can see where they come up with the terminology. Now, we want to replicate DNA. So in the nucleus of the cell, DNA starts to unzip to make its nucleotides available for other free nucleotides in the cell. Now, when it unzips, it's using an enzyme to break. These are hydrogen bonds that we talked about earlier in the semester. They're strong enough to hold it together, weak enough that we can break them up and make some more. So in the nucleus, there's free nucleotides. Here's a thymine comes by and attaches to the adenine. Here's a guanine comes by and attaches to the cytosine. Adenine comes by and attaches to that thymine. Same thing on the other side. Adenine to the thymine, cytosine to the guanine, and a thymine to the adenine. And we have 2 brand new strands of DNA that look just like the first strand, just like the original mother strand. These we'll call replication, obviously. This is happening in interphase of the cell cycle. Remember the G1, S, and G2 of the cell cycle of interphase? S. That's it. DNA synthesis. S. All right, now on the test, I will give you a sequence. Let's say I give you A, C, T, G, T, A, C, A, G. And I'll tell you, I ask you on the test, multiple choice question. Match this DNA sequence. So on your choices over there, you're gonna look for a choice that starts with what? A T. And next would be, finish it out. There it is. That's the one you're looking for, because A and T always matches and C and G always matches. If I ask you to match an RNA from this DNA sequence, what would match it?
U, G.
Well, y'all are all over this one.
U,
Thank you. We at least
G.
get the U right, don't we?
U, C.
U again, and a C right there. So what we did is replace the thymine with a uracil. In RNA, that does not use thymine, it uses uracil in its place. So even if you couldn't remember it, do another DNA, wipe out the T's and put a U in there and you've got it. If you just remember adenine and thymine always join, cytosine and guanine always join, in RNA, U replaces T, that's it. It's as simple as that. That's what it takes for replication. Now, if we move to transcription, we've replicated DNA in the interphase of cell cycle, S part of interphase. Now to transcribe, we're unzipping in the nucleus, but this time we used another enzyme to do it with called an RNA polymerase. So let's say we have the same thing. We have A, we have a C, we have a T, and here's an A, a G, and a T. But this time, we're going to use one side or the other as the template. Because RNA is a single strand, DNA is a double helix. Being a single strand, we just need one side or the other. So here we would match up a U, correct? Here we'd match up what, a G? And here we'd match up an A. And that strand there, once it's made, would actually unzip itself from the DNA. That's actually a messenger RNA, is what it is. Made in the nucleus. Once it's made, it unzips itself and travels out to the cytoplasm where it joins to a ribosome. What's our ribosome definition from first test? Cytoprotein synthesis. So we've already transcribed the messenger RNA. Now we're out in the cytosol, or out in the cytoplasm, to translate a protein. So far so good? Not too bad? All right. We're going to carry the code. This is messenger RNA carrying a U, a G, and an A. Out here, there are transfer RNAs. Let me draw this. Transfer RNAs are considered the anti-codon. The messenger RNA is the codon, and that's represented in threes. It's a triplet code. Now let's talk about this for a minute. How many amino acids do we need to make a protein? 20. 20 is your magic number. 4 is not enough, 16 is not enough. If each one of these bases represents an amino acid, we'd only have 4. If we used combinations of 2 of these amino acids, we could get as many as 16. We're still lacking. So we have to use combinations of 3. This is what we figured out over time with technology. There has to be combinations of 3 used, which gives us way more than 20. Gives us a whopping 64 combinations. Well, that's a little overkill, isn't it? This is what it does. It gives you margin for error. Now, if you think about that, margin for error, I'm going to show you how that applies in just a minute. So let's link this up. let's create a protein chain, and I'll show you this margin for error. What's matching that U right there? Very good, here, and here. Would you agree that matches? Okay, that creates an amino acid. The amino acid's left dangling off the ribosome. That transfer RNA gets out of the way, and another one comes through, and this chain is way out here. It finds three more and it creates another one. It gets out of the way. Some books even call those taxis. It comes through, finds another one, leaves another one. Now the shortest chain here is 5 that we know of. The average polypeptide chain or amino acid chain that makes a protein is 500. And it takes less than 20 seconds for the cell to do it. It's really efficient. It will reach a point out here where these three may say stop or end chain. When it says that, it's finished, and whatever it's built, and in the sequence it's built, is the protein that it has produced. Look on page 89. On page 89, you see a purple box down at the bottom. You see up the left-hand side, you see UCAG. Across the top, you see UCAG. And down the right-hand side, you see several sequences of that. And when you match them up, if you match the first one up, you get U. That's an amino acid called phenylalanine. We could tell stories about phenylalanine. There's actually been movies made because of lack of phenylalanine in people's bodies. It actually creates a substance in newborns lacking the amino acid phenylalanine. It's a genetically inherited trait called phenylketonuria, and it makes their blood turn to almost antifreeze and it kills them. So, needless to say, notice there's UUU and UUC that'll give you phenylalanine. Two different combinations. Look at leucine. Leucine is UUA all the way down to CUG. Well, there's several combinations that'll give you that. Isoleucine, there's 3. Methionine, there's 2 or 1. Valine, there's several. Okay, let's say, find on there glutamic acid. GAA or GAG. Well, let's just use GAG. That's a good one. GAG represents glutamic acid. Let's say that we were translating the protein hemoglobin for your red blood cells. And in this sequence of proteins down here, a sequence of amino acids, let's say glutamic acid, GAG, was supposed to be right there. But instead, somewhere down this line, we got off one little unit. It'd been easy here. It could have been easy way back here, except DNA checks itself. But we got off one. And instead of GAG, look, where's valine? Look, instead of GAG, let's do GUG. GUG. Instead of GAG for glutamic acid to give us hemoglobin, we got GUG, we got valine instead of glutamic acid, and now instead of hemoglobin, we have sickle cell anemia. That's all it takes. But, look at this. In glutamic acid, what if we'd have made one mistake? Instead of GAG, we got GAA. Would that make a difference in the end results? It was still glutamic acid, wasn't it? We still got hemoglobin, and we're still normal. There's your margin for error. Can mistakes occur? Yeah, those are mutations. Changes in DNA are mutations. How many mistakes have occurred that still got you the same amino acid, that still got you the same protein, and we'll never know? There's a question we probably don't want to answer. That'd freak you out, find out how many times we screw up. Every new generation has at least one new mutation, whether good or bad. Keep in mind, animals don't tolerate mutations very well. So they gotta be acceptable or you wouldn't be alive. In this case, that's kind of a null and void mutation, isn't it? We got the same results. That's your margin for error. Any questions at this point on replication, transcription, translation? The main things you need to be able to do is know those definitions and match some bases. If you can match bases and know those definitions, how many of y'all plan on going on to physiology? Eventually you'll be in physiology. But if you're in my physiology, there'll be questions just like you get in here on that test, just off this stuff here. I'll step it up a little bit off these. I'll have a question like in replication. I'll say, Joe has a physical ailment in which, for some reason, his body's producing too much DNA. The doctor has developed a medication that slows his DNA production back to normal. What is it slowing? Is it slowing replication, transcription, or translation? Replication. So we already know the definition, let's apply it. I mean, that's all it is. And sometimes I'll make up a name of a drug that's never been written in any book. And I get people, they'll take that test and they'll say, how are we supposed to answer that question? We're not pharmacists. We've never seen that drug. We've never heard of that drug. How are we supposed to answer that? Well, read the question. It tells you what the drug does, okay? And I mean, I've had a lot of gripes and fusses, but I'm trying to prepare you ahead for that. The last thing in this chapter deals with a little bit of oncogenes and tumor suppressor genes and things like that. And now on page 96 is where we're at. And it's basically telling you just a little bit of research they've done. Cancer cells, we already know we inherit those. Those can be inherited. We can develop them. They're called oncogenes. Hopefully those are turned off. Most are alive. If they're turned on, cancer's going to develop. Tumor suppressor genes, that's part of our immune system that if it finds something like that growing out of control, it can actually go in and destroy, seek, kill. Those pretty nice genes to have. Oncogenes occur normally in cells, and in their normal form, they are called proto-oncogenes, or they're for cancer. It's unfortunate that we have these, but they're there. Down at the bottom, they talk about P53 and P21. I think we discussed this a little bit in an earlier page, it's physiology book, it's page 74. Have we talked about these two genes, P53 and P21? Well, don't let me repeat myself. If I have, tell me. But I'll tell you what they are. These two genes influence mitosis. These two genes play a role in producing a substance called cyclin. Cyclin tells the cell to mitotically divide. So in their research with laboratory animals, they went in with x-rays and they bombarded, they killed the P53 gene. When they killed it, mitosis ran rampant. The cyclins were produced in enormous amounts and mitosis ran rampant. But what do we have if we have mitosis out of control? Cancer. Exactly what it is. So they went in and they looked at the P21 gene, which was closely associated with P53, and what they ultimately found out, that if either of the genes were destroyed, or if both of the genes were destroyed, that animal developed cancer. No questions asked. Now, have we done anything with it to fix cancer? To my knowledge, no. We're just a little smarter on how cancer gets started. We can't stop it yet. We can give you chemo and kill some more cells, or radiation and make you miserable and kick in that ball gene real fast with radiation, can't we? And that's all they're talking about down at the bottom. All right, so that finishes that chapter. Now, in your notes, you have another page of notes in your notes in reference to chapter 37, correct? What is today? Thursday? We're shooting for a test next Thursday. We like tests on Thursday, right? Bad time, last time. Okay. Well, I'll probably lecture ahead a little bit Tuesday, but we'll review Tuesday. But let's look at these notes today. We'll review Tuesday, prepare for the test on Thursday. That sets up good for the other class as well. All right, if everybody's looking at that, I'm just gonna go through these. You don't even have to go to chapter 37. If you'll study these notes, the questions are almost verbatim in your study guide. If you'll start working on it between now and Tuesday, you'll see there's just a lot of statements on there and a lot of stuff right off this piece of paper. So but please work that up before Tuesday so you can ask me questions. Number one, the biosphere is where all life is confined. If it can live, it's underneath the biosphere. Biomes, these are based on plant distribution. Different environments are categorized or separated out because of the types of plants that's growing there. If it's a tropical rainforest, it's called tropical rainforest because of the type of plants that's growing there. If it's a desert, it's because of the type of plants that's growing there, okay? The greenhouse effect is increase of CO2 due primarily to burning of fossil fuels. Ecosystem is the highest level system consisting of both living and non-living organisms. A community, an assemblage of species sharing the same environment. That could be a variety of species: mice, ducks, geese, chickens, dogs, cats, whatever. A population is how many there is of that particular organism, how many ducks there are, how many mice there are, how many pigeons there are. An organism is the individual of the species. You're the organism, a single organism of a human species. Ecology is the study of the relationship, and it should say the relationship of the organism and its environment. Niche is your place in society. This is what you do best. Everybody has specialties, everybody has gifts. You won't ever catch me in an art department drawing anything. I think y'all can vouch for that. You've seen my drawings, right? My little stick men that change shape every time I redraw them. They're a little warped when I get finished. That's not my niche. A producer is the first step of the grazing food chain. It's green plants. Now, we made the statement earlier in the semester, green plants are primary producers, and they utilize sun where we can't. But we like the green plants. We like a little salad with our beef, right? Consumers are the final step. They're heterotrophs. We're a consumer. Decomposers, fungi and bacteria, that kind of finishes off everything right there. Energy flow begins with photosynthesis and it uses visible light. That's sunlight. That's what it is. Herbivores, second step in the grazing food chain. They're eating the plants. Carnivores are meat eaters. So they kind of fall in there as kind of a third step. Maybe not a final step necessarily, but at least a third step. And then here's some biomes. Temperate deciduous forest. They tend to shed their leaves on an annual basis, and they tend to have a variety of animals that live in them. Oak and maple are examples. Where would you find a temperate deciduous forest? Maybe I need to require botany ahead of this class.
Here.
Exactly. Bodo, Oklahoma is a temperate deciduous forest. Exactly right. Now, there's a little typo on the next one on coniferous forest, because I put pine, it's supposed to be pine, fir, and spruce. Instead it says pine and furry spruce. But we gotta get a chuckle out of it, so why fix that? That's my question. There's no need fixing that. You'll remember furry spruce. And then you'll put together, oh yeah, Mr. Wally, a little bit nutty, but that was a fir and a spruce. Where do you find this particular type of forest? We're not, there are a lot of these trees here, yeah. The firs and the spruce don't do well here, but pine, we do well with pine. We're gonna be up north. This is made for long, cold, snowy winters. We didn't have that last winter. It was pretty mild. But just long, cold, snowy winters up north. Tropical rainforest, remember it has the greatest variety of both plants and animals, yet the soil is not fertile at all. They thought, man, look at all the plants growing. Holy smokes, we can wipe out some of these trees and make us a little garden spot here, and we'll just grow enough stuff, we'll stop world famine. No, all they did is let it wash off into the local river because there's about that much soil, and the only thing that was holding it was the plants that were there. So, yeah, man's not the smartest person in the world sometimes. Not very fertile. Grassland. Rain falls too scant to support trees, but not enough to prevent a desert. Is there grassland in Oklahoma? Kind of between here and way west or Panhandle, you get in lots of grassland. They're not desert by any means. In fact, Oklahoma probably has the greatest diversity of biomes than any other state. Texas would be number 2. We go from far southeast corner of Oklahoma. Anybody from down around Annabelle? Close to Annabelle? Have you ever heard of Annabelle? Okay, yeah, I know you hadn't heard of Tom, Oklahoma then. You have heard of Tom, Oklahoma. This is like a spot in the road in the far southeast corner, and it's got a swamp around it. So that's as low as you get in Oklahoma elevation, right there, all the way out west, and you come through the mountains to get there. Now, is that not difference in biomes? If you go to Guyman, I consider Guyman a desert. Although it's still a grassland, that's about as desert as I'll ever see, probably. But there's your difference in biomes. The desert, let's see, the tundra. Let's look at the tundra. Short growing season, specializes in severe cold climatic regions, dwarf woody plants, grasses, and lichens. And then the desert, plants are adapting for conserving water. Now, you're gonna get into your cacti here. Animals have nocturnal habits. Man, there's just not much rain in the desert. Cold nights, nocturnal habits, because it's so hot during the day. If they come out during the day, things will dehydrate. The plants have such thick, waxy leaves that they only breathe at night. They can't breathe during the day, or they lose too much moisture. And that's the difference in day and night in the desert. And I asked several questions about the biomes. They're on the study guide. And the last one, nitrogen-fixing bacteria converts nitrogen into nitrates and ammonia. You will find nitrogen-fixing bacteria with plants that are called legumes. Name me a legume. What?
Alfalfa.
Beans, alfalfa, peas. The biggest one out there, soybeans, probably. But in our average pasture, any type of clover would be one. So, nitrogen-fixing bacteria, we have tried since the 60s to try to fix this bacteria to the roots of corn. Corn absolutely loves nitrogen. If we could get the corn to convert atmospheric nitrogen to its own nitrates, we would see fuel prices come way down. Because it's endless what we could produce, but we can't genetically make it happen yet. And I bet there's somebody still working on it, because they'll be wealthy if they ever come up with it. Any questions? Let's take a short break.